MCQs on History of Biotechnology. 1. The earliest evidence of biotechnology, such as bread and beer fermentation, dates back to around A. 2000 BC, B. 4000 BC, C. 6000 BC, D. 1000 BC. Explanation: the Sumerians and Babylonians practiced fermentation of cereals using yeast for beer production around 6000 BC, marking one of the earliest known uses of biotechnology. Correct answer: C. 6000 BC. 2. The ancient Egyptians contributed to biotechnology mainly through A. Genetic engineering, B. Bread and wine fermentation, C. Vaccine development, D. Antibiotic production. Explanation: Egyptians were among the first to use yeast in bread making and wine making, 4000 BC. Correct answer: B. Bread and wine fermentation. 3. Selective breeding of animals and plants to improve traits is considered a form of A. Modern biotechnology, B. Synthetic biology, C. Ancient biotechnology, D. Recombinant DNA technology. Explanation: selective breeding is an ancient practice, 2000 BC, used to improve yield and desirable traits in crops and livestock. Correct answer: C. Ancient biotechnology. 4. Who is regarded as the father of vaccination? A. Louis Pasteur, B. Edward Jenner, C. Robert Koch, D. Alexander Fleming. Explanation: Edward Jenner developed the first smallpox vaccine in 1796 using cowpox material. Correct answer: B. Edward Jenner. 5. Louis Pasteur's major contribution to biotechnology was A. Discovery of antibiotics, B. Recombinant DNA technology, C. Microbial fermentation and pasteurization, D. Discovery of PCR. Explanation: Pasteur showed that microbes cause fermentation and developed pasteurization to prevent spoilage. Correct answer: C. Microbial fermentation and pasteurization. 6. Gregor Mendel is best known for his discovery of A. Germ theory of disease, B. Laws of inheritance, C. DNA replication mechanism, D. Recombinant DNA. Explanation: Mendel's pea plant experiments, 1865, established fundamental principles of genetics. Correct answer: B. Laws of inheritance. 7. Which scientist developed pure culture techniques and established Koch's postulates? A. Louis Pasteur, B. Robert Koch, C. Alexander Fleming, D. Kary Mullis. Explanation: Koch developed pure culture methods and formulated Koch's postulates to link microbes with specific diseases. Correct answer: B. Robert Koch. 8. The discovery of penicillin was made in A. 1865, B. 1928, C. 1973, D. 1982. Explanation: Alexander Fleming discovered penicillin in 1928, leading to the era of antibiotics. Correct answer: B. 1928. 9. Who confirmed DNA as the hereditary material in 1944? A. Watson and Crick, B. Avery, MacLeod, McCarty, C. Rosalind Franklin, D. Frederick Sanger. Explanation: they demonstrated that DNA, not protein, is responsible for heredity. Correct answer: B. Avery, MacLeod, McCarty. 10. Watson and Crick proposed the double helix model of DNA in A. 1944, B. 1953, C. 1962, D. 1973. Explanation: with X-ray data from Rosalind Franklin, Watson, and Crick discovered DNA's double helix structure in 1953. Correct answer, B. 1953. 11. The invention of recombinant DNA technology, rDNA, is credited to A. Cohen and Boyer, B. Sanger, C. Mullis, D. Doudna and Charpentier. Explanation: in 1973, Stanley Cohen and Herbert Boyer inserted foreign DNA into plasmids, founding genetic engineering. Correct answer, A. Cohen and Boyer. 12. Frederick Sanger is associated with A. PCR, B. DNA sequencing, C. Cloning of Dolly, D. CRISPR discovery. Explanation: in 1977, Frederick Sanger developed the chain termination, dideoxy, method of sequencing. Correct answer, B. DNA sequencing. 13. The first recombinant DNA drug approved for use was A. Penicillin, B. Humulin, insulin, C. mRNA vaccine, D. Interferon. Explanation: in 1982, humulin, human insulin produced in E. coli, became the first recombinant DNA drug. Correct answer, B. Humulin, insulin. 14. PCR, polymerase chain reaction, was invented by A. Kary Mullis, B. Watson and Crick, C. Rosalind Franklin, D. Stanley Cohen. Explanation: Kary Mullis developed PCR in 1983, enabling DNA amplification in vitro. Correct answer, A. Kary Mullis. 15. Which organism's genome was sequenced first? A. E. coli, B. Haemophilus influenza, C. Saccharomyces cerevisiae, D. Arabidopsis thaliana. Explanation: in 1995, H. influenza became the first organism with a fully sequenced genome. Correct answer, B. Haemophilus influenza. 16. Dolly the sheep was cloned by A. Ian Wilmut and colleagues, B. Herbert Boyer, C. Edward Jenner, D. Craig Venter. Explanation: in 1997, Dolly became the first mammal cloned using somatic cell nuclear transfer. Correct answer, A. Ian Wilmut and colleagues. 17. The Human Genome Project was completed in A. 1990, B. 1997, C. 2003, D. 2012. Explanation: the Human Genome Project began in 1990 and was completed in 2003, sequencing the entire human genome. Correct answer, C. 2003. 18. CRISPR-Cas9 technology was introduced by A. Watson and Crick, B. Jennifer Doudna and Emmanuel Charpentier, C. Frederick Sanger, D. Robert Koch. Explanation: in 2012, they demonstrated CRISPR-Cas9 as a genome editing tool. Correct answer, B. Jennifer Doudna and Emmanuel Charpentier. 19. The first mRNA vaccines, COVID-19, were developed by A. Moderna and BioNTech, B. Pfizer and GlaxoSmithKline, C. Roche and Novartis, D. Pasteur Institute. Explanation: both companies developed mRNA vaccines against COVID-19 in 2020. Correct answer: A. Moderna and BioNTech. 20. Which of the following is not part of ancient biotechnology? A. Bread fermentation. B. Yogurt production. C. Selective breeding. D. Recombinant insulin. Explanation: recombinant insulin belongs to modern biotechnology, developed in 1982. Correct answer: D. Recombinant insulin. 21. Robert Koch's work laid the foundation of A. Genetics. B. Germ theory of disease. C. Recombinant DNA. D. Synthetic biology. Explanation: Koch's postulates linked microbes to specific diseases. Correct answer: B. Germ theory of disease. 22. Which discovery directly led to the concept of genetic engineering? A. DNA double helix. B. Recombinant DNA technology. C. Pure culture technique. D. Antibiotics. Explanation: Cohen and Boyer's rDNA work in 1973 allowed scientists to modify genetic material directly. Correct answer: B. Recombinant DNA technology. 23. The first clinical trial of gene therapy was conducted in A. 1973. B. 1983. C. 1990. D. 2000. Explanation: the first successful gene therapy was for severe combined immunodeficiency, SCID. Correct answer: C. 1990. 24. Which of the following pairs is correctly matched? A. Fleming, DNA sequencing. B. Avery, penicillin. C. Jenner, smallpox vaccine. D. Mollis, fermentation. Explanation: Jenner pioneered vaccination in 1796. The other options are mismatched. Correct answer: C. Jenner, smallpox vaccine. 25. Which of the following best represents the chronological order of discoveries? A. Mendel rightward arrow, Jenner rightward arrow, Crispr rightward arrow, Pasteur. B. Jenner rightward arrow, Mendel rightward arrow, Pasteur rightward arrow, Fleming. C. Fleming rightward arrow, Mendel rightward arrow, Watson rightward arrow, Boyer. D. Boyer rightward arrow, Pasteur rightward arrow, Mollis rightward arrow, Doudna. Explanation: Jenner, 1796, vaccine. Mendel, 1865, genetics. Pasteur, 1857 to 1876, fermentation, germ theory. Fleming, 1928, antibiotics. Correct answer: B.